Thanks, Rick, and thank you to Fraser. I think he must have stepped out a second, but thanks for the opportunity to talk. If you're here in the morning, you already heard a lot about connective tissue disorders and Chiari malformation, and you're about to hear more. Oops, I don't know what happened here. It looks like there were, yeah, I don't know what happened.
What do you, hold up.
I don't know,
Switch one of the things, you see.
Oh, yeah, I think it's that one, yeah.
Okay, so
I don't know why it didn't.
You want to the end?
No.
You want the up there?
It was supposed to be up there.
All right.
Yeah, I don't know what happened.
Okay, let's see.
Okay, well, we'll wing it. Some of the slides somehow are a little off.
Outline.
Here we go.
Good.
Perfect. Thanks, Paolo. So this is the outline of my talk. First of all, I'm going to review a little bit about hereditary disorders of connective tissue. And I'm not going to review Chiari because you guys already know about Chiari, but just to talk about the subset of the patients who have both conditions. And then I'm going to move on and talk a little about the linkage analysis that our group did and how we showed it's important to take into account some of these co-occurring conditions in the genetic analyses. And then I'll just conclude. So hereditary disorders of connective tissue disorder, or connective tissue, really are a group of medical conditions that are characterized by defects in the extracellular matrix. And this includes mutations in genes which form collagens, elastins, and fibrillins. So I'll talk about more specifically some of the specific disorders, but a number of these genes are known. There's mutations which have been identified in a number of these. Overall, the frequency of these conditions is fairly rare. It's about 1 in 5,000 to 1 in 10,000. Both males and females are affected. And a lot of these conditions do appear to be sporadic in nature, but there's a lot of examples of familial forms. And when you do have a familial form, it's almost exclusively autosomal dominantly inherited. So I've listed here some of the more common connective tissue disorders. Ehlers-Danlos is one of those. Clinically, even amongst Ehlers-Danlos, it's very heterogeneous. There's a number of different subtypes. I'm not going to spend the time to talk about all these different subtypes, but there's a number of genes which have been implicated. They're primary collagen genes or proteins that make post-translational modifications to the collagens. So, and then for Marfan syndrome, this can be clinically distinguished from Ehlers-Danlos or EDS because it has chest abnormalities, and these individuals also tend to have long limbs. So Marfan's is caused by mutations in fibrillin. And then there's also Loeys-Dietz Syndrome. And this is further clinically distinguished by aortic aneurysms, hypertelorism, bifid uvula, and sometimes even cleft palate. So this group is even more genetically heterogeneous by mutations in the TGF-beta pathway. So in two of the receptors, TGF-beta 2, which is one of the ligands, and SMAD3, which interacts with this pathway. So right off the bat, even amongst the connective tissue disorders, you see that there's a tremendous amount of clinical as well as genetic heterogeneity. You know, and perhaps I'll stop right here and say, even amongst our own patients, we have patients who have a history of a number of these different conditions. So before we started our genome screen, we actually sat down and talked about, should we screen specifically for these genes? One of the challenges is, especially for ENES, those collagen genes are extremely repetitive. So we did have a little bit of experience in the lab attempting to sequence through some collagens. Wasn't the most pleasant experience because they are so repetitive. So we actually elected not to screen those. But I will say that was by Sanger's sequencing, not next-gen sequencing. So we might have better luck with newer technology. So, you know, Hal already showed some of these, but clinically, you know, we can pick these patients out because of the hypermobility. They have the stretchy skin, you know, there's all these sort of circus performers you'll see with all their stretchy skin and how they can move. But they also have scoliosis, and scoliosis often is a little bit of a more subtle comorbid condition that goes along with Chiari, and we don't often talk about that so much. And then there are sometimes these vascular abnormalities, which can range from something as simple as bruising to something which is much more significant, such as aortic dilation. So moving on and talking about Chiari malformations, you know, Millerad and a number of you guys in the room remember he has kind of characterized a number of different subtypes or kind of maybe underlying mechanisms leading to Chiari malformations. The most common, of course, is the cranial constriction, which that one supposedly is due to the small posterior fossa and the cramping. But we know among the Chiari patients, you can look in the literature, there's a number of different sort of co-occurring genetic syndromes. And this is a huge sort of overwhelming laundry list of those co-occurring syndromes. However, what strikes me when I look at this list, I show in red a number of these conditions really are truly connective tissue disorders. And of course, in bold are some of the more common. So Hal already sort of pointed everyone to Millerad's 2000 paper, which is a beautiful paper. You know, and really at this point, probably the best and the only description we have for the co-occurrence of these connective tissue disorders in Chiari, and really what is their presentation and how does it compare to patients who don't have connective tissue disorders. So in their analysis, and this wasn't, and they admitted, this is not a true epidemiologic analysis. This was a case series. So amongst not quite 3,000 patients, what they saw was about 13% of the cases had a co-occurring connective tissue disorder. So if you put that frequency together with the frequency of Chiari malformations, we're looking at about 0.1% of the population, so pretty infrequent. But some of the other distinguishing characteristics of these patients were they had a younger age of onset, they had a greater female to male ratio, which I think is interesting and not sure why that happens, particularly because there is evidence for autosomal dominant inheritance, so this doesn't completely fit. There's also a higher incidence of a number of different clinical presentations, lower brainstem abnormalities, hypoplasia of the oropharynx, reduction in the clivus axis, clivus atlas, and axis atlas angles, which, everyone sort of talked about already this morning. And then, like I just mentioned, there's evidence for autosomal dominant inheritance. And I will say, just anecdotally, amongst our own patients, looking at these pedigrees, the patients who have connective tissue disorders, it really is much more clear that there is some kind of an autosomal dominant pattern compared to sort of the classic Chiaris. There's some evidence for autosomal dominant inheritance in those patients as well. It just isn't quite as obvious. So I pulled table 1 from that paper just to show you what were the specific types of connective tissue disorders. By far and large, it's EDS, mostly hypermobile EDS, but there was also some Marfan-like, and then some others, which included osteogenesis imperfecta. So it's a laundry bucket of connective tissue disorders. But I think if we can go back to this slide, this might be the classic Chiari, but this may be Chiari plus connective tissue disorder. So now I'm going to move on and transition into genetics because that's what I do as a geneticist. So we already knew a priori, Chiari is very complex. There's got to be multiple genes, multiple environmental factors involved. So our group set out to do a linkage screen. And these were our criteria. We had to have at least 2 family members who met criteria. But importantly, for the rest of the interpretation of our results, we actually excluded folks who had formal diagnoses of EDS and Marfan's. So we had actually taken those folks out of our analysis. And then for DNA, we extracted it from blood, we had MRIs, and then we had a lot of information about co-occurring conditions. In terms of the genetics, we genotyped over 600,000 SNPs across the whole genome. And then we did a linkage analysis in a couple of different ways. So here was the results. This was for all families. So I want to point you here to this line above 3. So that's kind of the magic cut point when you're doing linkage analysis to declare significance. So here we had a number of different hits with a number of different statistical approaches. And these are just single point analyses. So what that means is we're just looking at a single SNP to see if that single SNP predicts Chiari. So that's what this is. Then we went on and we did what we call multipoint analysis, because if you're truly in a region of linkage, you should be able to see that if you have one marker that's associated, the markers that are closest to it should also be associated. And so if you do a multipoint analysis, you would expect the odds score should increase. So this, again, is with all our families together. So we do have a few interesting regions. But at the end of the day, while there were good biologic candidates, the scores really just didn't increase that much. So we really didn't feel confident about starting to embark on sequencing and follow-up of certain candidates because we just didn't have a whole lot of evidence for specific regions. So for that reason, then we sort of kind of changed what we were doing. So, and this is just summarizing, based on the limited significance of those results, we decided we would start stratifying the families. So the idea would be, here's our total population, but if we stratify them by the clinical characteristics, perhaps we'll have improved power to detect a gene in one group and a different gene in a second group. So this is where it gets interesting. We did exclude folks who had a formal connective tissue disorder diagnosis, but despite that, we still had a lot of families who had symptoms which were consistent with the connective tissue dysmorphology. So they didn't necessarily meet the formal diagnosis, but they had some of these symptoms, such as hypermobility, mitral valve prolapse, aneurysm, kyphosis, etc. So as luck would have it, when we stratified the families who had these symptoms, those that did and didn't, it essentially split the data set in half, which was good for statistical power. So now I'm presenting the connective tissue positive families versus connective tissue negative. So I think you can appreciate, and these are the 2-point results, you can see that now we're actually getting different hits. We're getting different hits in the positive families versus the negatives. So then we moved on and we did our multipoint analyses. And this time we actually had a little better success with the multipoint analyses. So for the connective tissue positive families, we had a couple of hits on chromosome 1. We had some kind of interesting regions on chromosome 9. And then in the connective tissue negative families, we had a nice peak on chromosome 8 and chromosome 12. So I'm going to focus on those regions. So at this point, we have not yet done sequencing in these families in these regions, but I wanted to highlight for you where those 2 peaks are on chromosome 1 in the connective tissue positive families. There's a bunch of interesting candidates. So here's this PBX1 gene, which is shown to be expressed in bone-derived cells and osteoblasts. So it appears to have some kind of a role in osteogenesis. DDR2, this is another gene also in that region. And mice who are knocked out for this gene have this interesting skeletal phenotype, and they were able to show that this reduction in growth is due to chondrocyte proliferation rather than kind of cell death. And interestingly, too, when they culture the fibroblasts from those adult mice, they see they proliferate slowly, and they also have a diminished wound response. Then there's this other gene, this PRRX1, where mice who were homozygous for the mutant allele died soon after birth and exhibited problems in skeletal structure development. So while we haven't sequenced these genes yet, they're actually really great candidates. But the one that I'm actually most excited about is at the bottom. Hopefully you can see it. It's TGF-beta 2. So this is in the more distal region on chromosome 1. And the reason I'm interested in that gene is mutations in TGF-beta 2 have been found in families with Loeys-Dietz Syndrome. So, which again is one of the well-known connective tissue disorders. So that's where we are with connective tissue positive families. Well, I did, just real quick, I wanted to tell you a little more about another approach we took. So before, everything I showed you was the presence or absence of Chiari malformation. So now we're taking a little more quantitative approach. So we had a bunch of MRI measures. We created these composite traits with principal component analysis. We showed they were heritable, and then what we decided to do is see if we could use these traits, again, to improve our evidence for linkage. So this is how it works. So we'll have these families, we'll order the families based on the size of the posterior fossa height. You can see low to high. And then what we do is we just keep adding up the evidence for linkage. And so what happens is at some point you will find an ideal subset of the whole data set, which gives you the maximum peak. So that's what we're attempting to do. And so when we did this, again, with posterior fossa height, we got a significant Loeys score over 3, and this was on chromosome 22. So, and just so you know, this included about 40% of the families. And also to point out, this was the original Loeys score. So see how much the Loeys score increased when we focused just on those families who had high posterior fossa height. So one of the candidates in this region we were excited about is this EP300 gene. So it's involved in chondrocyte differentiation, and it's part of this bigger complex which inhibits collagen 2A1. So, and more interestingly, this gene, as well as CREB-BP, which it kind of interacts with, they both have been implicated in Rubinstein-Taybi syndrome. So, and I showed you the chromosome 22 peak where EP300 was, but interestingly, in the region where CREB-BP is located, we also have a little bit of an interesting peak. So, and this is just to show you how those genes work together. So, I may be going over time just a little bit, but for a moment to talk about connective tissue negative families. So the two regions we found in that subset were chromosome 8 and chromosome 12. And we were really excited when we saw that because both those regions harbor genes part of the growth differentiation factor family. So GDF6 on chromosome 8 and GDF3 on chromosome 12. So mutations in those genes had previously been associated with Klippel Feil-Syndrome. And we know that maybe as much as 5% of Chiari patients also have Klippel Feil. So these seem like great candidates. So when we sequenced those genes, we did end up finding mutations in GDF6. So this mutation in particular is what we picked up. And it is a complex mutation. We know it's got reduced penetrance because not only is it associated with skeletal phenotypes, but it's been previously reported to have ocular phenotypes as well. And one of the challenges that we saw as well in our own families, it did not segregate purely in our families. So you can see here the folks with the mutation, those are the folks who have, that carry this particular mutation, but this individual here does not meet criteria for Chiari malformation. And then conversely, there's this aunt up here who does not have that mutation, but she meets criteria for Chiari malformation. So I think you can just see how complex this is. So I suppose to interpret it and to step back a little bit, what I think is probably happening is there's multiple genes that are contributing, and maybe in this family, this particular mutation contributes and modifies risk, but it isn't necessary and sufficient for risk. So, this is just to summarize. So far, this has been the largest linkage screen to date. I know a number of different groups are starting to work on genetics as well. But importantly, we did find that stratifying on connective tissue symptoms and phenotypes really did make a huge difference in finding specific genomic regions. These are the two papers that data came from, in case you're interested. So just to pull it all back together, I think that we all know at this point there's a large portion of the patients that have these co-occurring connective tissue disorder symptoms, as well as Chiari. And I think we probably need to start characterizing those symptoms a little bit better because for you guys as clinicians, it certainly may make differences in how you treat them. But I believe very strongly, based on the data we have so far, that this is also important mechanistically. So genetically, these patients are different. So, and I'll stop there.
Do you postulate that there are multiple abnormalities in the same person, but they have to be on the same chromosome, don't they?
No, not, sorry, let me get back to the mic. No, not necessarily. Like, I can give you an example. We have a neural tube family where it's an anencephaly family. They had 3 anencephalic pregnancies, and they finally, God bless them, had a normal child. And when we did exome sequencing on that family, we never found one mutation which completely co-segregated with all 3 affected Instead, what we found was a burden of mutations that was higher amongst those 3 affected siblings that was part of the PCP pathway, the planar cell polarity pathway, compared to their normal sibling. So you might want to think of it more like a threshold, excuse me, a threshold effect. So there's probably multiple mutations which are contributing. That's part of the reason this is just so hard. I mean, even when we did the stratification, I think you saw we had multiple linkage peaks, suggesting that even among the more homogeneous clinical group, we're still getting evidence for multiple genes. So I think that this is really, really complex. You know, and one of the things, as I mentioned in the beginning, we did not screen the known collagen genes and fibrillin and that kind of stuff. So I think it would be really interesting to go back and do that as well. You know, I just think it's really, really complicated. Yeah.
Do the candidate genes that you have meet the requirement, or if you know in the literature there's a description of them acting as dominant genes?
So the genes themselves, so, you know, a lot of the supportive data that I put up was from animal models, mouse models, so forth. So those were knockouts, right? So, yeah, so in that particular context, sorry, my throat's starting to get dry. In that particular context, sometimes you saw heterozygote knockouts with phenotypes. Sometimes it required, you know, a homozygote knockout. You know, for me, and, you know, the two of us have talked a lot about animal models and how to do those in human genetics. I think we need to find the human mutation first. You know, we have to find the human mutation first, then we can move on to the zebrafish, the mice, and so forth, model those, see what they're doing functionally, and then that will really help us make sense. Is this a dominant mutation, or is it recessive?